it's my pleasure to introduce Professor Wim Vranken, who is at the Free University of Brussels. Wim started off in chemistry at the University of Ghent in Belgium before moving on to conformational analysis of HIV peptides using NMR, and continued on with NMR analysis of peptides and proteins when he moved to McGill University in Montreal. And then subsequently what was then called the Biotechnology Research Institute in Montreal. Then during a sort of a gap year at the Free University, he rediscovered computer programming and started to move solidly in that direction of computational aspects of NMR. And in fact, that's the context in which many of you have known Wim. He's been a collaborator with BMRB over the years. He's been a constant presence at the Gordon Conference on computational aspects of biomolecular NMR. So for many of you, Wim is an old friend. He spent the core of his work over the past decade or so at the Protein Data Bank Europe and the European Bioinformatics Institute in Hingston, which is just outside Cambridge in the UK. But he's now back currently a research professor at the Free University, the Vrije University Brussels, I guess would be something closer to the correct pronunciation in Belgium. And he's closely associated with the IB2 Inter-University Institute of Bioinformatics in Brussels, which he is the former director. Wim, thank you so much for agreeing to do this. And I also want to point out that Wim has offered to make this just the first of a series of talks on a range of topics that I know will be of very broad interest to this group. So Wim, thanks for joining us, and look forward to your presentation this morning.
All right, thank you very much, Jeff. Yeah, so I'm going to be talking about the work we did on which was published a couple of years ago on auto-encoding NMR chemical shifts from their native vector space to a residue-level biophysical index, or shift crypt for short. Now, I don't think I have to explain to this audience what chemical shifts are. I mean, you all know that it depends on the shielding of the atoms, which chemical shift value they have, which frequency they resonate at, and this resonance frequency is of course normalized and to give a chemical shift value expressed in ppm. Also, I think I don't need to explain that when your protein is unfolded, it has many different conformations that are in quick dynamic exchange. You get chemical shift values that are very close to each other because each of these atoms experience the same average environment. Whereas if you have a folded protein, then your chemical shift values are more spread out because each atom in each, even if it's in an alanine, of course, has a very different environment because of the fold of the protein. In between these extremes, you have the middle range as well, where in principle you can detect from chemical shifts as well which conformations your residue adopts on average, and also how much it moves. So chemical shifts have been used again. This is well known, of course, for together with information from X-ray structures mostly, to estimate very precisely per amino acid what the phi angles are, and programs like Talos. There are other ones out there as well, of course. Basically what you do there is you relate the chemical shift value for well-folded proteins to what you see in the conformations that you see in those well-folded proteins, often from X-ray structures, to then very precisely define the phi angles associated with those chemical shift values, because they represent the conformation of that residue. On the other hand, for heterogeneous conformations, methods have also been developed. So the idea here is, okay, we see the average of the chemical shift value over a certain time span, like sort of low milliseconds. We can use that information to then also estimate what is happening to that residue, and then get characteristics for each amino acid in the protein, not quite 5 psi level, but more how much is this residue moving? So the backbone dynamics, that was the RCI developed by Wishart and Baryansky in Canada. And then sidechain dynamics in Edmonton, I think, right? Wishart. Then also the sidechain dynamics. So that was a follow-up of that program. And also things like secondary structure population, where I'm just showing the method that I was involved in Cambridge with Michele Vendruscolo, Delta 2D. There, the idea is that based on this chemical shift information, you can make an estimate of like the population of alpha helix or beta sheet that you might have, because there could be some interchange. You could have 90% helix, 10% unfolding in a protein, for example. So 1 second. Okay. So the idea is you can use chemical shifts for well-folded protein to get quite precise estimates of 5 psi values in the case of programs like Talos, but you can also use it to get more general broad estimates of what might be happening to that residue, even when the protein is moving quite a lot. So what we did now already 8 years ago is to generate a set of curated chemical shifts from the biomass bank, on which we applied the random coil index. Yeah, so when I say curated, I mean like we made sure it was in a certain temperature range, pH range. There was no other agents in there like urea or something that would induce unfolding. And we got basically an estimation of backbone dynamics using the RCI for just over 2,000 proteins, constituting about 220,000 residues, which encompasses more or less the average behavior of those particular proteins in solution. But importantly, also that training set included completely disordered proteins and also proteins that were like, molten globule state. So you get in that set are proteins from fully folded to completely disordered in essence, and everything in between, which is quite different from methods in structural bioinformatics trained on folded proteins from the PDB, of course, because there, you need to have one fixed conformation in order to see that. So what we did basically is to use that, develop a method then where you could sort of get an idea of the general dynamics, if you want, of a protein. So this is less accurate, clearly, because the RCI is not very accurate, but it does give you a sense of what the residue is doing. And importantly, like I said, it includes IDPs, loops, and it basically captures motions up to the low millisecond time scale because of the chemical shifts. So in fact, what you do then is you can relate these dynamics, not very accurate, but still, to the sequences of those 2,000 proteins. And then we used machine learning to relate backbone dynamics to the sequence and developed this method called Dynamine, where you basically input a sequence and you get as output a prediction of the backbone dynamics for that particular sequence. So this has been around for a while. I say backbone dynamics, but this is slightly confusing because actually it was related to the S2 value, So basically 1 means that it's rigid and 0 means that it's very flexible. So it's the RCI converted into the S2 order parameter, where 1 is full order and 0 is complete disorder. So high values mean rigid, low values mean flexible. So basically, we've played around a lot with this method to try and understand what it actually is doing, where it works and where it doesn't work. And it made us really think about revisiting this estimation from chemical shift data. So in essence, can we do better than methods like RCI? Because really what you want is to try and account as much as possible for dynamics and conformations. And also by that, after 5-6 years, there was more chemical shift data becoming available, especially, of course, for intrinsically disordered proteins, because that's really where NMR can deliver results that no other, traditional structural biology technique can give a lot of information. Also, of course, by now there's more structures available, also more molecular dynamics information, new force fields for IDPs, yeah, that can better represent the motions and conformations of these IDPs. And of course, also now we have vast improvements in machine learning. And when we developed Dynamine, neural networks were sort of, well, they were already in use, but not that common yet. We used a very simple linear regression, basically, approach. Now we have very complex neural networks that in principle should be able to capture much better what the protein is doing. I think everyone knows about the recent results from Google in the protein structure prediction. Clearly, there's a lot of improvement possible there as well. So in essence, the methods that we'd been using until then, delta to D for the secondary structure population, are models. Yeah? So the delta to D uses protein structure information to determine the average shifts that you would get for helix and sheet, and then use a Lorentzian model that employs these average shifts and considers each atom type separately, and they are then combined in essence. So, okay, like does this H alpha say whether it's helix or sheet, the C alpha, and then they combine, or these different values are combined together. Whereas RCI for dynamics uses as a baseline the secondary chemical shift, so the difference from the experimentally determined chemical shifts from GGXGG, where G is glycine and X is any amino acids, random coil peptides. So it uses the values for the X for the 20 amino acids as a reference point, and then looks at the difference from it. So they also in there use a linear model that combined these secondary chemical shifts that was tuned on molecular dynamics information. But again, in essence, each atom type is considered separately and then combined, in this case, by using a sum value. So one is essentially structure-based, yeah, as a reference point. The other is random coil peptide-based. But there are problems with both of these approaches if you think about it, because the structure-based one has clearly inherent bias in the data. It only considers well-folded proteins, where there might be, that might not reflect very well what proteins are doing when they are in fact quite dynamic and moving between different conformations. And then also, as we all know, the NMR structure calculation process is flawed, yeah, where we basically try and represent one model to average NMR data, which never quite works the way it should. And X-ray structures especially do not represent well what might happen in solution, in terms, especially in terms of like, you know, populations of secondary structure. Yeah, I mean, typically still X-rays at cryogenic temperatures. It's been shown in previous papers that the movements that you might see from the X-ray structures don't actually represent very well what the dynamics are of that protein in solution at room temperature or 310 Kelvin. On the other hand, the random coil-based approaches are maybe also not the best, because if you look at different peptide series, then they will have different chemical shift values. So GGXGG or GGXAGG, for example, or QQXQQ will give you different reference points. And that indicates, again, thinking about what chemical shifts are, that these peptides do have very different inherent conformation preferences. Was there a question? Because if so, stop me, yeah? I can, all right. Also, there have been molecular dynamics studies, extensive ones, that point to inherent conformational bias of amino acids. So even conformationally heterogeneous peptides. So there could still be a bias of an alanine or a glycine or a glutamic acid in these GGXGG peptides that actually then affects the reference points that used to determine the secondary chemical shift value. Someone is producing some noise, background noise, I think. Is it? It might be Frank. I think maybe you're not muted. Okay. Anyway, it's very difficult to decide from this what is actually the best approach. Yeah, using the structure, using random coil information. Both have biases in essence. So also what's important to consider with chemical shifts is that they co-vary. Yeah, like chemical shift values within one amino acid are interconnected. Yeah, just to show you this simple 2D plot where you have here the H alpha chemical shift and here the C alpha chemical shift for alanines, I think in this case. So each point represents the chemical shift value for one alanine extracted from the bioMcRAS bank, related to the conformation that you see in the structure, where red is strands, blue is helix, and green is a coil, and yellow is other. So you can see that there's like a clear progression, but there's also clearly a path in there. Yeah, like certain combinations are not possible. Yeah. And there is a path in the interconnection between these two chemical shift values. So it is also important. Yeah. So here, for example, as well, you can see that there's almost no variation anymore in the C alpha, but there is in the H alpha, and that probably means something. Yeah, with respect to the behavior of that residue in that protein. So if you don't consider the correlation between these different chemical shift values, so the hyperdimensional picture, if you want, then you will lose information if you don't consider all of these at the same time. Now, here you might think, okay, I've got the C alpha, so it's always the same, but it's not really, because there's also a change in the H alpha that means something. And then here you have a very different pattern in essence. So you need to get the most out of this information. You need, in essence, a multidimensional approach using all the different chemical shift dimensions. So in this case, H and C alpha, H alpha, C and C beta, typically, because those are the ones that are the most commonly available. But current models, like I said, delta to the RCI don't do that. They consider each shift separately and then sort of add up everything. And we as humans can only grasp 3 dimensions, right? So hence the machine learning comes in. Also, what's important to think about is if we analyze that kind of information based on our preconceived notions about what the chemical shifts mean in relation to structures, which is very folded protein based, I would say, then we are restricting ourselves to a certain extent, because chemical shifts are complex data. They encompass both dynamics and conformation, and we don't necessarily really know, exactly how that works or whether we can get more information out of there. So can we use machine learning or artificial intelligence, whatever you want to call it, to analyze this multidimensional space? The advantage of using an agnostic approach, where you basically don't assume anything about the data, is that you don't need any reference values, you don't need to use any structure information, you don't need to use reference peptides. Machine learning can easily operate in an arbitrary number of dimensions. There are no problem for the computer to do that, even if we don't understand what it's doing really. So why not try and interpret shifts in their native multidimensional per amino acid space? That was the thinking. So we basically only assume, the only assumption that we make in essence is that this chemical shift path that I trace in 2 dimensions exists in this hyperdimensional space somehow, and that the path that you see for each amino acid has a similar meaning in biophysical terms. So if you are at one point for an alanine or at one point for a tyrosine, the extreme end of this path, it means rigid alpha helix. If you are at the other end, it means rigid beta sheet. And then there's a path that sort of has a similar meaning in terms of the biophysical behavior of the residue. So first we created a new chemical shift data set from the bioMcRAS bank. So like I said, we make sure that we limit the variation environmentally. So pH between 5.7, temperature between 293, 313 Kelvin, no order or disorder inducing agents, so no urea, no TFE, that kind of thing. Chemical shifts for 3 for H, C, and N atom types. NMR structures available as well. In this case, for to have a reference set that we could compare to. And then we also applied chemical shift re-referencing to this data set to make sure that everything was correctly or at least referenced in the same way. And then also we removed top and bottom 1% outliers because there's bound to be always some issues in there with the possible paramagnetic sites. I mean, we did try and take that out, but just to make sure that we were not including the extreme data points, which often are also wrong, right? I mean, that's still, there's still errors in there. So we then ended up with a curated chemical shift data set for almost 3,400 proteins. So clean, this is in principle is clean and dependable data. And then we proceeded developing this shift grip method. And this is a neural network that does encoding, decoding, developed by a former PhD student, Gabriele Orlando, in the lab. That was published 2 years ago now. So the essence of this approach is that, so you have all the shift data per amino acid. So we have all the data points bunched together for alanine, for like cysteine, for aspartic acids, for glutamic acid, and so forth. Yeah, we have all these chemical shift values. You then basically have a neural network that tries and encodes the inputs normalized chemical shift values into one single number. Yeah, so it learns how it can compress all this information about the chemical shift values into one single number. And then what you do is with so that's the encoding part. And then with the decoding part, basically, you then try and predict again the original chemical shift data from the single number. Yeah. So what this neural network does in essence is to try and find the best encoding in this one single value of your original chemical shift so that it can then decode them again to get, you know, the original chemical shift values back. The idea there is that you try and find a number, yeah, that best sort of captures the variation that you have in the original chemical shift values, so you can then also reproduce them by decoding them later on. In essence, so what you get is you start off with scaled chemical shifts from 0 to 1. You end up with a single value per residue, and then you try and, like, you know, you can then recompute the original chemical shift values from this single value, and that's the concept. So, I mean, obviously, I mean, this is all good and well, but then what actually do we get out of these chemical shift values? Or these shift group values, at least? So how do they relate to structure-based parameters, yeah, for which we had information from NMR? And we only use NMR structures here because they, you know, even though and then when we calculate NMR structures, we make one model based on, you know, like one model that tries to capture, in essence, average data. Still, in principle, some of the variation that you might have in, for example, secondary structure population might reflect itself in these models because there will be less information if you only have like 70% helix or something. So that's the general idea. We ran strides to get 5 psi angles for a helix, 5 psi angles on helix sheet coil designation for these residues, and then related them back to the shift grip values. So then here you have the shift grip index value at the bottom, and then you have the distribution of the residues that were designated as helix, coil, or sheet by stride in these NMR structures. And you can see that, helix is on one end, sheet is on the other end, and coil is in the middle. So in essence, values of 0 mean helix, values of 1 mean sheet, and values of in the middle mean coil, probably dynamic. Yeah, so that's good. At least that, you know, it could capture basically what we know of protein behavior just by you know, completely hands-off learning on the chemical shift data. You can do that a little bit more in a little bit more detail. So if you look at the 5 psi angles then related to these shift grip values, if you look at the first 16 percentile, so that's the lowest 16% of the shift grip values in the data set. You can also, the data points here are colored, so each dot is a residue. I mean, it's hard to see here, but yeah, they're colored by the amino acid type. You can see that they're really the core alpha helix region. Yeah. If you start to go, if you take the next 16%, yeah, then you can see that you get also some data for other areas in the 5-side plots. If you then take the next 16%, then yeah, the residues are quite distributed over the Ramakamran plot. Even more so when you, yeah, but still there's a preference somehow for the helical here. If you go even further, then yeah, it's really all over the place if you want. It then starts to, with the next percentile, starts to group in the beta strand region, and then you get like, yeah, reasonably well-defined beta strand for the final, the top 16% of the values. So that's good because it, again, it shows that it captures somehow the conformation of proteins, even though it was completely hands-off learning. You can also look within each class that was designated from the structure. So if you only look at the helical residues, you can see that the low shift crypt values, the ones that are the closest to 0, actually encompass the central alpha helix region, so really the core alpha helical region, whereas the higher ones sort of are more spread out, surrounding alpha helical region, if you want to call it that. For coil, you have, again, the low regions are more alpha helical in nature, whereas the high values are more beta sheets or strand centered. In sheet, again, you have like a region here for the high values that is core beta strand, if you want to call it that. And then you have a red region, which is sort of surrounding beta strand, so not quite as sharply defined. So that's good because it shows that it's capturing somehow some of this also probably dynamic behavior if you think about the structures in a statistical way at least. Other things you can check are, for example, how does it relate to solvent accessibility? We already showed that like more than 10 years ago that exposed atoms in amino acids tend to have narrower chemical shift distribution and tend to be coil, which is sort of what you would expect intuitively. This is again that same plot that I showed you earlier for the H-alpha, C-alpha here, but now colored by exposure. And you can see that the exposed residues are like, you know, have a very similar H-alpha shift, but actually can have quite a wide distribution of C-alpha shifts, for example. The fully beta strand and alpha helical ones are buried in essence, or tend to be buried. Okay, so if you look now at the relationship between shift grip index versus solvent accessibility, you can see that, and here we basically classify based on whether they are exposed or buried, yeah, and then we plot the distribution of the shift grip values for those categories, and you can see that for the exposed, you do have the exposed residues in alpha helixes, there is a shift towards the middle to 0.5, whereas the buried ones tend to be a bit lower, but the difference is not that big. Significant though. Here you've got for coil, yeah, the exposed ones and the buried ones also have a slightly different distribution. And then for beta sheets, again, the buried beta sheet ones tend to be give higher values than the exposed ones. So everything towards the middle tends to be more exposed dynamic. Yeah, everything towards the edges tends to be more buried / rigid. We also compared it against early folding. So we also did some work on early folding earlier, where we had a database from residues that start to fold earliest in vitro folding from AGX experiments. So, you know, that showed where the protein is primed to form structure. We have a database for that as well. I'm not going to get into the details there, but if you compare now this shift grip index versus early folding residues, and you can see that actually for alpha helices, there's not really a difference between the early folding and the non-early folding. For coil, neither. For beta sheets, on the other hand, the early folding ones, which are the ones that are supposed to really, how should I put it, are locally primed to form. Like if you think about early folding, it's the residues that are close to each other in the sequence, so local interactions between amino acids, that are going to be important because there is no, no global fold yet. The idea is that if you have favorable contacts in the local sequence context, then that will be primed to form some sort of structure. Yeah? So that on the beta sheet side, this comes out on the alpha helical side less. Well, not really. But that's nice to see that there's such a big difference there. So it means that it really somehow captures this kind of behavior even in the final unfolded protein. Yeah? These residues tend to be more beta sheet and more rigid than other beta sheet residues that were designated beta sheet in the folded structure. Yeah, so there's still a difference there that you can capture somehow in this shift curve value. If that makes sense. That wasn't the best explanation, but I hope you're following me. If not, let me know. So what we also did is compare shift script versus the random coil index. So here you've got the chemical shift index value, so the shift script value versus the calculated backbone dynamics, RCI. What you would have expected is probably something like this, where, RCI high S2 values, yeah, for a full-on beta strand and full-on alpha helix, and then coil dynamics for residues in the middle here at 0.5. There is also, however, a whole region here that RCI says is quite rigid, that we say is around 0.5. Yeah, so I'm not sure what that means. What is good though is that you can clearly see that you have this like the down peak here at 0.5, which means that for the fully dynamic residues we're getting the same results. Not so for other residues. So it's hard to really establish what is going on there because we don't necessarily have the data to really validate that independently. But as often, the proof is a sort of, and to me at least, a lot of understanding of how a method works comes from data that is like, not, or at least not directly related to what it is that you're using. So what we did in essence is to see if we could use this to compare the behavior of different proteins. So here you've got two proteins, a strawberry and a cherry allergen. They have the same folds and 79% sequence identity with each other. I mean, they're very easy to align, as you can see, because they're almost the same. Yeah. So if you look at the shift script value for these two proteins, where in red you can see all the residues that have been that are different between the two. Yeah. So the red lines here mean that at those positions you have a different amino acids. Yeah. You can see that the pattern that you get in shift script values is very similar, even in positions where you have different amino acids. Yeah. So it means, going back to what I said earlier, that the shift script value seems to capture the biophysical behavior, if you want, of a residue independent of residue type. And that's an important point, I think. So it's sort of, average out the or at least captures the essence of the biophysical behavior of amino acids, irregardless of whether it's an alanine or a tyrosine or whatever. So that was nice to see, of course. If you look at delta 2D, of course, you also see similar patterns, but you can clearly see here that you don't get the fine-grained patterns that we see in the shift curve values in the same way, right? This is much more like it's helix or it's not helix, right? Whereas here you really get more of a fine-grained pattern that aligns quite well, even though the proteins are only 80% similar in terms of sequence. This is the same, but for beta, a similar picture, I would say, for delta 2D. We went further as well with this. So here's a thyroredoxin 1 from different organisms with 46% sequence identity. Again, very similar folds. Here you can see the, so one's from Taliani Raffaellavicea. You can see here the sequences, and in red I've indicated where the sequences are different, and in black it's where the residues are conserved. If you look at the chemical shift values of the two, one is in dotted line, the other one is the red line. You can see that there are similarities, yeah, for C alpha, C beta, H alpha, but you know, it's hard to say where these are more similar or more different from each other. For the NH values, you get this. Again, there's similarities clearly, but you know, not necessarily that easy to compare, to see where it's more the same or less the same. I mean, people obviously have used methods like, you know, for HNs, for example, the H and then times a certain value plus the difference in N value and things like that. But I mean, where that comes from is not clear, because as you could also see from the path in the 2D plot, one change in chemical shift value, yeah, it changed from like 53 to 52 is not necessarily the same as a change from 49 to 48, for example, right? For a C alpha, say, it can have a very different meaning. And that's not something that you can capture if you just take the difference, of course, in these cases. So here we get the shift crypt index, yeah, for these two proteins. You can see that it's very similar, but there are some notable differences, like here and here, for example. Interestingly, though, if you look at the regions where you have highly similar values and differences, you can see that for this stretch that I've indicated here by the blue box, the first bit, the sequence is completely different, yeah, but the shift crypt index is actually almost exactly the same. Whereas here, you have like a proline and then 3 other residues. You do seem to have quite different behavior between these two proteins. And then here, for example, you've got a part that is exactly the same in sequence, but there seems to be actually a difference in the biophysical behavior, if you want, in the protein in this particular location. And that we've related that back to the structure as well in the article. I'm not going to get into detail there, but I mean, it's interesting that it seems to capture differences at the biophysical level, if you want, in between these proteins, and similarities as well, of course. We also had developed a version that is sort of a more a light version based on the HN and C alpha changes for changes in a free and bound form of RIM-M with the S19. And you can see here that, yeah, again, here it captures differences between the bound and the free form. And we also showed in this case that this kind of information complements the traditional approaches to detect changes in the approaches that are based on just looking at the absolute difference in chemical shift value, in essence. Again, because we think that, you know, one change in chemical shift is not the same as another change in chemical shift in a way. And then finally, what we also did is look at the random coil peptides. So here you have different series of peptides, yeah, that we then interpreted with the shift crypt value. This is the GGXGG series from Schwarzinger in 8 molar urea, pH 2.3. Yeah. So you can see here the amino acids and then the shift crypt value that they give. Yeah, so here, for example, so this is the beta strand, and if you want, this is more the alpha helical end. You can see valine threonine, yeah, especially gives like high beta strand regions. So that would indicate that threonine already has a very strong bias for beta sheet conformation. So if you look at differences for threonines, they don't necessarily mean alpha helix, they just mean less beta sheet in a way, yeah, if you interpret it that way. If you then look at the values for Kierkegaard from also XGGX, but in 1 molar urea, they're quite similar with similar trends, but there are some notable differences, like here for aspartic acid. That's probably because of the pH difference. Yeah. Then this GGXAGG series, also in 8 molar urea, yeah, you can see gives very similar results, yeah, quite consistent overall. A GGXPGG series then gives notable difference for example, here for valine and also for proline. Yeah, so I mean, clearly this proline has a bigger effect on some residues than on others. And then here, if you look at the QQXQQ series, which has been used, I think, a lot for intrinsically disordered proteins, no urea, you can see that everything is basically shifted to the beta region, if you want, of the shift crypt space. And interestingly, this is being used a lot to show, that there might be some alpha helical conformations. But if you sort of turn the argument around and say, okay, how does that influence the basic conformational preferences, then you can see that they seem to shift to the beta strand region. So that also means that you would tend to see, if you use this as a reference point, you would tend to see a lot more chemical shift values that indicate some kind of alpha helical tendency, because obviously, your reference point shifts to the top in this case. Yeah.
Are these results sensitive to the salt concentration?
This I do not know. I didn't put the salt concentration, but I'm assuming they could have an effect. I'm not sure if there's a consistent chemical shift series for salts that anyone has done, but that would be interesting. I mean, I'm sure there's lots of other stuff going on. What's interesting, I think, about this is that even though obviously I'm not saying that the shift crypt value is the holy grail in this kind of approach, it is unbiased in a way. And it would be interesting to see, at least relatively speaking, how conformational preferences change for different amino acids. You could get that information out in principle, I think. But we've not gone further than this. So anyway, there's lots of things that one could try with this, like IDPs, obviously. Do they have behave similarly with different amino acid sequences? Chemical shift series, like you just said, with salt, but also with temperature, of course, like to see how temperature might affect the intrinsic conformational bias of an amino acid. We also have not done, unfortunately, comparison to extensively studied proteins like ubiquitin, for which there is obviously lots of relaxation and dynamics data. In principle, apply this method to RNA and DNA as well. What we have developed in the meantime, which is also available from our server, is an alignment procedure where you can basically use the shift crypt value to align proteins, either only based on shift crypt value or based on shift crypt value in combination with the sequence. So that could be also quite interesting to sort of biophysically align, if you want, proteins, which is also something that we're interested in. Aligning proteins by intrinsic characteristics of residues, not by the sequence in essence. So it seems to act like the shift crypt index as a kind of a summary of the conformation and maybe dynamics of amino acids and proteins, sort of a biophysical index. You inevitably lose detail, but it provides sort of a distillation of the chemical shifts in a human interpretable form, instead of looking at 5 dimensions at the same time and then deciding this is more important than that. And the machine learning AI approaches are basically only as biased as the data that they use. Yeah. So if you have reasonably unbiased data, then actually they provide a less biased view. This is in relation, of course, to machine learning being used in for example, face scanning, which is typically dominated by white Western males, as I'm sure you all know. Machine learning in itself is not biased; it's data that's biased, right? So in a way, it's less biased if you can use it that way. And then just briefly to finish, I'm just going to give a quick overview of other things that we do in the lab so that you have an idea as well. So we have these sequence-based predictions not only for backbone dynamics, but also for sidechain dynamics and for conformational preferences. This is still based on RCI and delta 2D. We have an early folding predictor as well, a fault mine, that tells you where in your sequence you have where your sequence is likely to start folding the first. And so determine possibly also the folding pathway to a certain extent. We have a disorder predictor as well, which combines information from different other predictors. An aggregation one, and all these three in essence some use sequence, some don't. Like eFoldMine doesn't use the sequence at all. It uses only the biophysical characteristics that we predict from sequence. So in a way, it's more generic, because it doesn't look at the sequence. And we tried incorporating sequence in the training, but that didn't work because you have quite a small data set. It worked the best when we worked off predicted biophysical properties to predict yet another property in essence. Agmata uses a model as well that's based on biophysical features. Disomine also uses partially these biophysical features. We also have done some work on phase separation protein prediction, PSPR, based on an HMM-like approach. We've also been working quite a lot with people that do very state-of-the-art analysis of mass spectrometry data, and then Gemachtens in Ghent. And they can determine the location of PTMs and many different PTMs. There's now also a project that just started where someone is working on, again, proteomics and mass spec-based the subcellular location of proteins. And now we're trying to combine the information about PTMs, subcellular location, the interactions that the proteins have, also depending on subcellular location, together with biophysical characteristics and the effect that PTMs might have on the biophysical behavior of the protein, for example. So that's quite exciting. We just started that this year. I've also been working on the prediction of deleteriousness of amino acid variants in human proteins. That's the Diogen 2 project, Mutaframe. That's continuing as well, now more in relation to cancer. This one was more rare diseases. And then also, this is something that I'm interested in, is protein design. I think some of the methods that we develop can be useful for protein design because you capture something that is in between sequence and structure, if you want. I think that ShiftCrypt shows that as well, is the design of these megabodies where you couple these nanobodies to a large protein to then use it in electron microscopy. So the website has all the information, and we also now, this is just in review now in our nucleic acid research web server, we've got a one-stop prediction shop where you can upload either sequence files and multiple sequence alignments or chemical NMR chemical shift data to get shift grip information and all kinds of other data. That's a one-stop shop for upload. And yeah, so this work was done by Gabriele mostly. Also Daniele was involved. They both are now doing a postdoc in Leuven. And the website I've mentioned, and this is mostly funded by the FWO, which is the Flemish Regional Funding Institute. So that's it for me. Any questions?